of July 1996, a sheep named Dolly was cloned from its mother's genes. The two look exactly like each other, and this was done with recombinant DNA, specifically cloning. But what is cloning? In biology, DNA cloning is a technique used to make many identical copies of a piece of DNA. However, it doesn't just mean recreating an entire whole organism, it could also just make multiple copies of DNA. To make it clear, let's talk about how it is done. First, DNA will be extracted from a chosen organism and cut by a restriction enzyme. After the gene is cut, it will be placed on a plasmid and will be put together by an enzyme called ligase. Additionally, gene from antibiotic resistance will also be added to the plasmid. This will be explained further later. Next, the bacterial transformation process will be done. This is where the gene bound to the plasmid will be introduced to a bacteria and given a high temperature shock to encourage the plasmid to take up the foreign DNA. Now, the sample that contains the bacteria will be placed on a plate with nutrients and an antibiotic to be cultured. However, not all the bacteria in the sample solution will be taken in the gene, meaning that all bacteria, even ones not containing the gene, will grow on the plate, making it hard to identify which one contains it. This is why an antibiotic is also placed on the plate. Now, remember the antibiotic resistance gene added to the plasmid earlier? This will be helping the bacteria with the gene survive. This way, bacteria not containing the gene will not grow. Then the bacteria will reproduce and create multiple copies of the gene you wanted to clone. Finally, you've completed your cloning. Definitely, there are many benefits this new technology can achieve. One of them benefits the field of pharmaceuticals. Specifically, it helps with the production of insulin for diabetes and the human growth hormone, which is used as a treatment to prevent strokes. Now let us all recall. Cloning is an rDNA technology done to create multiple exact copies of a gene with the help of a restriction enzyme and ligase. Then these are cultured with the help of a bacteria that will do the production of multiple copies of this gene. After all that you've learned, let me ask you, do you think this type of technology should be accessible to everyone, or should there be a limit to who can use this type of technology?